First, we have Associate Professor Dr. Syarul Nutakain Baharum from University Kebangsaan Malaysia. Speaker 2, we have Dr. Arif Izairi Zamani, also from University Kebangsaan Malaysia. Last but not least, we have Dr. Nizar Hapiana as Speaker 3 from Bandung Institute of Technology in Organic Chemistry Division. And this webinar is moderated by Mr. Andi Rifki Rosandi, PhD, from University Center of Excellence for Nutraceuticals. Before we start the webinar, I would like to remind you to fill the questionnaire and attendance form on the given link to claim your certificate, and we will issue the certificate after the webinar is over. Well, first, we will hear our welcome speech from Associate Professor Al-Fahmi as Head of University Center of Excellence for Nutraceutical.
Okay, thank you very much, Mrs. Nurinanda Rizky Komaladewi. Very good morning, everyone. First of all, I would like to welcome you in this seminar, webinars, mass spectrophotometers and NMR spectrophotometry in metabolomics. How to start your own research. It's our great pleasure to tell you that this webinar dedicated to our researchers around Indonesia, of course, also around, I think, the Asian country. Maybe also the participants come from the other outside of Asian country to learn, to learn, or maybe to nurture the network about the research on the application of spectrophotometry and NMR in the metabolomic. And we know that the development of science and technology, especially the computer aid with the information technology, is very satisfied and help us to solve many problems in our research, of course, in our life. And today, University Center of Excellence, Bioscience and Biotechnology Research Centers in ITB, organizing this webinar, and we would like to share from our speaker. We have already our speaker now, this young speaker, of course, some of them, and then for expert, they have expertise in mass spectrophotometers, and can be useful to do the metabolomic studies. I would like to thank Associate Professor Dr. Syarul Nutakain Baharum from Symbiosis University Kebangsaan Malaysia, who would accept our invitation to be the speaker for this seminar. And then also Dr. Arif Izairi Zamani, also from Symbiosis University Kebangsaan Malaysia. And of course, our rising stars, what I would say is our millennial researcher, Dr. Nizar Hapiana, Organic Chemistry and Division, Institute of Technology, Bandung. Everyone, I would like also to thank our moderator today, Mr. Andi Rifki Rosani, PhD. This is our excellent research in our groups, and also in the PUI nutraceutical, and also of course for master ceremony, and then I think also the people behind this, Dr. Agus Cahyadi, Ari Ranwers, and our teams, Ola, and all organizing committee who participated to organize this very nice seminar. And of course, I would like to thank also to Peter Berka, right? BNM. I'm really familiar with this one because we use a lot of Agilent product during my study as well. Also now in our research. Thank you for your support for this seminar. And of course, seminars not only just to share our knowledge about mass and NMR spectrophotometry in metabolomic. And then we will learn from our expertise today. But I would like to really emphasize my message now that we can do a lot of work in research, and especially in natural products. And we know in Indonesia we have a lot of biodiversity, biodiversity that can be developed for the health purposes, for global health, also for the welfare. So maybe after this seminar, we have keep in contact and also continue our network, our collaborations to give the good impact, the best impact for the research. For the research, not only about to produce our the output for the scientific paper, but also the output for the product, for example, that can be used by people, by communities to improve the health status. So this is what I would like to emphasize to you. So I think without any further ado, I would like to officially open this webinar. With this saying Bismillahirrahmanirrahim, the mass and NMR spectrometry in metabolomic, how to start your own research officially open, and hopefully that all participants can get advantage, can benefit for this one, also improve the metabolomics. And I believe that the participants come from the different background, right? Maybe some of you are just new coming for this topic. Some of you might be already expert with this one. Let me discuss in this seminar. Thank you very much for your attention. Hopefully this seminar will give us a good benefit for all aspects, of course. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Thank you, Dr. Al-Fahmi, for the welcome speech. Next, I will introduce to our moderator for today's webinar, Mr. Andi Rifki Rosandi, PhD. He currently works in University Center of Excellence for Nutraceuticals as researcher. He expertise in chromatography and spectrophotometer, structure and illustration. He is also an expert in the formulation of essential oils. All right, now we move to the main session. So please welcome Mr. Andi to run the main session.
Okay, thank you, Ms. Nurin. Firstly, I will introduce our first speaker, Associate Professor Dr. Syarul Notakain Baharum from Institute of System Biology or INBIOSIS, University Kebangsaan Malaysia. Her expertise is basically in metabolomics, the field of system biology. Is the study of grouper fish, Alzheimer's disease in human, and she is also an expert in several spectroscopy methods. Dr. Ain here is to present the topic mass spectrometry strategies in metabolomic case study. Dr. Ain, you may now start the talk. Thank you.
Thank you, Dr. Andi Rifki, for the nice introduction. So I would like to thank Institute Technology Bandung, especially under the Center of Excellence Nutraceutical, for the invitation to me to talk to all of you, participants from Indonesia. I believe there is some from Thailand and also from Malaysia as well. So I would like to start my presentation by giving you a brief introduction. Oops. On the introduction to the metabolomics, okay? So the topic that we'll talk today on the mass spectrometry strategies in metabolomic, a case study. So I'm from Institute of System Biology, University Kebangsaan Malaysia. So I'm sure most of you know about UKM. So you can go and visit our website on the INBIOSIS to see what is our current research development. So first of all, I would like to give you some information on the publication trends in the metabolomics field. So this is the based on our book. We have published a book on the omics application for system biology. You can get this book through the Amazons, and if you want to buy it from us, let me know. Okay, this is a publication trend since 2000 to 2018, talking about the metabolomics. The keyword that we use here is the metabolomics in article, abstract, and keyword. So you can find here, there is a lot of publications in very high impact journals, Q1 and top 10% journals mostly. So from the frontier in plant science, bioanalysis, analytical, chemical, acta, and metabolomics journal as well. So this is the trend on the publication since 2014 to 2020, basically focusing on the keyword of human disease, cancer, degenerative, and also on the medicinal plant, cancer, degenerative. So you can see here, there is also a very good high increasing trend on the metabolomics field from the publication. So it's still increasing, and I'm sure that we are people that are working in metabolomics, this is a very good chance, or this is a very good advantage for all of us can publish our work in these kind of journals. Okay, I'm sure everyone know about this. This is the center of dogma from genomics into the transcriptomics, proteomics, and metabolomics. But for today's session, I will just talk about the metabolomics. Okay, so metabolomics is actually the most emerging technologies that are available nowadays, basically based on the omics data. So this has been studied like since 2000, I can say that. So there is a lot of study has been done on metabolomics, especially on the metabolic response, on the different traits, different significant physiology, and also on the bioactivity, and also any study on that biochemical pathway. So metabolomics also has been used, is very mainly used by mainly possible by using analytical tools such as the mass spectrometry. So what is a metabolomics? It's like a snapshot showing how the compound are present at the relative level at the specific time point. So you can see that there is a metabolites changes during the specific time points. For example, if you have the plant that grow in the morning, produce some smells, but at the night, maybe this plant doesn't produce the smell anymore. This is some kind of biochemical reaction that's happened in the plants. So there's a specific time frame for some of the compounds that we produce in the plant, for example. So metabolomics also can refer as a holistic analytical approach that cannot be guided by the specific hypothesis. Actually, we can't really put some hypothesis that says that this plant must produce this kind of compound at this time, or maybe during the mature stage or something. There is the other factors that also involved in this compound to produce, especially the environmental factors. So maybe the rainy season, the dry season, the drought season, and so on. So as I said, metabolomic tools is we use a lot of the analytical instruments in a metabolomic study. So this is the example of metabolomics system that been used in our labs in BIOSYS UKM. I'm sure you also have this kind of instruments in ITB. That but still, if you would like to use our instrument, you are welcome to use. So what this metabolomics advantage over other omics? I'm sure most of you, some of you may be working on different omics, maybe genomics, transcriptomics, maybe proteomics, but why we want to focus on metabolomics. Okay, why metabolomics is a current trend in everywhere in the world? Because of the information that we will get or we can cater from the metabolomics is the information that are very useful for in understanding the biochemical synthetic pathway. For example, if study on the disease trait, the most of the study on the disease rate, you need to know about the genomics, the data of the genomes or the genes, maybe more than 4,000 genes. Then there is a lot of other genomes data is not known. same as transcriptomy, you need to have about 150,000 transcript to understand what actually happened in that biochemical pathway. Is similar to the proteomics for the translation from the transcriptomic. But for the metabolomics, it's about like 30,000 compounds that that's already known, but there is still unknown compound. But for the to understand on the treat or on the disease, you need the information about these 3,000 compounds only to make you understand what is actually happened in that organism. Okay. There's two routes to metabolomics, the quantitative metabolomics and also the chemometric metabolomics. So for the quantitative metabolomics, this is like this is like a targeted method, okay, which is you're focusing on what kind of compound that you're looking for. Maybe you're looking for the secondary metabolites compound from volatile compounds, or maybe you are looking for the primary metabolites or something. So you have a targeted one, you have the standard automatic standard, so you can run together in your system, so you can identify the compound straightforward. Okay, this is very straightforward analysis. However, about the profiling or the chemometric techniques, you need to have the unbiased technique, or we can say is untargeted metabolomic strategy, which is you don't know actually what's actually the plant or the microbe produce what kind of compound. That's why you want to understand it, or you want to profile as much as possible compound that related to your study. This is, as I said, we're focusing on the untargeted metabolomics workflow, which is from experimental design to the sample preparation, okay, until the data processing, metabolite identification, and also experimental validation. So all this is untargeted metabolomics. So I'm sure that many of us are working on the natural product, especially in Indonesia and Malaysia, because we have a lot of biodiversity. So we are working a lot on the natural product type of work, maybe a different type of plant, maybe different type of marine microbial and so on. Okay, so this is actually what I can say is that metabolomic versus the natural product discovery is actually is a comprehensive and also complementary technology. Okay, you need to have that metabolomics people working together with the natural product people to come up with very groundbreaking discovery. Okay, that's what we have in our group. So I will talk about this later. Okay, so there is two flow. Okay, there's only two flow: natural product discovery and metabolomics workflow. Okay, so this is a common objective for metabolomics to identify. Okay, to identify what type of metabolites or compound that presence in the samples for the specific time relate to what type of biological question that you ask. Okay, but the main important thing is that the metabolomic is a bottom-up strategy for discovering a potential of bioactive compound. For example, when you do the natural product, you already have a target. Okay, this plant produce the anti-diabetic properties. This plant produce this antiviral properties. Okay, but then you do that natural product study, so you isolate the compound, so you come up with that biological activity and so on. But in metabolomic study, that will be a different way. So it's from the strategy with like you need to identify, you don't need to identify what type of biological activity that produced by the plant. Okay, you always can do the profiling, and then you see maybe one plant produce a lot of other biological activity. For example, maybe this kind of plant can produce antiviral, diabetic, and maybe also anti-cancer. So this is what we call it is a beautiful work of metabolomics approach. Okay, I'll jump into the study on my case study on the application of metabolomics in unraveling the potential of local herbs. Okay, so the study, we are focusing on the polygonal miners, or in Malaysia, we call it kesum. Okay, I'm not sure the name in Indonesia, but based on my previous discussion with the Prof. Sahidin from Halu Oleo University, he's mentioned that most of the polygonum in Indonesia doesn't have the smell as the one that we have in Malaysia. So that makes something interesting. Okay, so actually this plant is being used as in Malaysian dishes in the cooking because of the pungent smell. Okay. So this is a type of dishes. I hope that every one of you already have your breakfast. Okay, but normally these dishes we only eat during the lunch maybe or maybe dinner. Okay, not during the breakfast. I think. Okay, so this is the laksa or asam pedas. It's a Southeast Asia style food. So you can see that this is the kesum compound. Sorry, the kesum plant. Okay, the kesum leaf. Okay, here in the dishes. Okay, why we want to focus on the polygonominous? Because of abundance of the oil glands. Okay, so this is can be as a potential in the production of essential oil. There's a lot of market industry on the essential oil available nowadays, especially because people are talking about the aromatherapy and so on by using the essential oil. So there's a lot of market for this plant to be used in the future. So this plant also produce aliphatic aldehyde, so it can be used as a food additive and also in the perfumery industry. Some old folk people using it as postnatal tonics, okay, and then it can be used in anti-ulcer, anti-inflammatory, and also in the cognitive enhancing activities. So as I said, because they have a very nice smell, pungent smell, so this been used as herbs or a spice in the food. Okay. So the idea of the project actually to demonstrate the use of the metabolomics approach in the model plant. So we know, every one of us know that what is the modern model plant. So Arabidopsis, right? So all the people in the world are using on the working on this model plant, which is Arabidopsis. Okay, we don't have the Arabidopsis in Malaysia. Okay, we also don't have it in maybe Indonesia. But why I want to focus on the polygonominous? Because this plant produce a lot of secondary metabolites that can be manipulated and also can be discovered for the potential of neurotherapeutic discovery. Okay, this is an overview the study that have been done in at Embiosis. So we have done on the transcriptomic, proteomics, genomics, and also metabolomics. So I'll be focusing on metabolomics because this is my part of the work. Okay, the metabolomics pipeline. So okay, this is what we have done in the polygonominous. So you can see here, a lot of effort has been done. Many years of study has been done on this polygonominous. We study on the volatile compound and also non-volatile compound. Even though this polygonominous produce a lot of volatile compound, but we do not discriminate the non-volatile compound because non-volatile compound also produce a lot of compound of interest. Okay, so this is all the techniques that we use for the study. Okay, I would like to share some of the publication that we have published in the work. Okay, this is the publication by using the GC TOF MS. Okay, this is a two-dimensional GC. Okay, I can say that not many places have this two-dimensional GC. So this is our advantage to produce the paper that time. Okay, so this by using this technology, we have demonstrate the powerful of the separation. Okay, between and identification of the compounds. For example, this complex compound, decanal and dodecanal. So you can see that the chemical structure. Okay, it's not much different actually, but it can be separated and identified by using the GCMS technology. So as I mentioned before, metabolomic also been influenced by the geographical distribution. Okay, so based on our study, we have tried to study on the different location. Okay, as we know that kesum produced at different location in the lowland and also in the highland. So in highland we have in Genting Highland and Fraser Hills. And also Cameron Highland, and then for the lowland we have the one that produced in in Ulu Yam and also in Lengging. So what we did is we want to control the environmental factor. So we try to make sure that there's not many variable for metabolomics, because once we have a lot of variable, that will be a problem for you to analyze your data. Okay. So basically, based on this study, we have put together all the plants, okay, based on different temperature and humidity, okay, in our growth chamber. Okay, so the study, part of the study has been done at the University of Sheffield, UK, when I doing my postdoc at that time. Okay, so this is the compound that we managed to identify by using this LCTOF. We have this toy in our place, okay, the LCTOF. So we have managed to identify the flavonoid and also other volatile compound by using by using the different distribution of metabolite based on the different temperature. Okay, so using this LCTOF, we have identified the flavonoid compounds, profile and identify. So you can see that's the location. Basically, this is the location from the original place. So we, as I mentioned, we took the plant from the lowland population and also on the highland population. You can see that the plants actually distribute or cluster based on their location or their environmental factors. Okay, but once we put it in the growth chamber and then we play around with the temperature, so the plant actually produce the compound of interest based on the phenotypics, okay, based on the treatment given to them. Okay, so you can see here, okay, this is the compound that produced by the baseline temperature, which is at the same temperature, the hydroxyl chalcone, and also at the lower temperature. This is all the biomarker compound that I mentioned to you here, okay, hydrocoumarin, and also at higher temperature is gallocalatin. All this is a flavonoid that very useful for the therapeutic study. Okay. So based on the distribution of the volatile compound, also we have found that based on the terpene compound, that the distribution of this plant basically difference between the species and also between of the location and also between the temperature that given to them. Okay, so for example, we produce a lot of other very nice compound like caryophyllene and farnesene at all the temperature, at all the location. Okay, but there is farnesene only produced at the low the temperature treatment. Meanwhile, the others produced at the higher temperature treatment. So you can see there's not much difference between the others compound. Okay, as I said, the compound have a biomarker as I mentioned before. Okay, so this is a classification based on the volatile compound according to the temperature. This is caryophyllene, basically produced at all the temperature, baseline temperature. And higher temperature, we produce the dodecanal, and at lower temperature, we produce that decanal and farnesene. So as I mentioned, dodecanal and decanal, not much different between the structure. Okay, there is a different on the a bit on the structure, but when they produce at different temperature, make them very distinct between each other. So we can from here we can manipulate the study. What kind of compound that you want more? Okay, for example, you want more dodecanal compared to decanal, then you need to grow them, grow the chromosome in a higher temperature, and so on. Okay, so from this different temperature treatment, we have managed to differentiate. Okay, the compound, the plant basically distribute based on their treatment given, not by their genotypic, but more to the phenotypic. Okay, this is the one that we mentioned before. Okay, so so this is another study by using the sensory evaluation for the flavor and fragrance for industry use. Okay, we have the GCMS system that have the olfactory detector. Okay, this is the olfactory detector. You can see here. So my student can smell the smell from that compound. Okay, and also from there we can run to the GCMS at the same time, and you see the peaks. and then you can identify the compound, and at the same time you can smell the compound. So it's something interesting, right? So we know that okay, farnesene smell like this, dodecanol smell like that, and other like other aldehyde also smell differently. Okay. So this study has been attract the attention from the Firmenich, one of the big company on the producing essential oil for the fragrance. Okay. So I would like to show you our kesum actually have the characteristic to be used as a perfumery industry because they have 3 different notes. As you know that perfume produce 3 different notes. Okay, for the lower part notes, the middle part note, and the last end notes. Okay, so kesum also show the same classification like that. Okay, so for example, I want to show here. This is the first note. Okay. Normally for the perfume industry, for the first note, what they want is the very strong smell, pungent smell. So we produce it, our plant kesum produce that kind of smell. So Nonana decanol have a very, very strong smell, pungent smell, very, what you call it, very strong smell, like smoke smell and so on. And then, okay, and then the second notes, okay, the second note should be the smell should be more nice smell or fresh smell. Okay, so our kesum produce dodecanol, cis-chirophyllene, humulene, and tetradeca, which have that orange smell, soapy smell. Okay, it's like a soap smell. Okay, and a bit a grassy smell. So grassy smell is more to like fresh smell, right? So and then the third note, okay, our kesum produce the fruity, floral, or woody smell. This is all the last note normally should be the pleasant smell. So and then last longer smell. So for the perfume, the very high quality perfume, they should have all these 3 different characteristics to produce the very, very expensive perfume. Okay, this is what I learned from the industry. Okay, so we are so lucky actually because kesum have produced all these 3 smell that make attract the perfumery industry to work with us. Okay, so based on different techniques, okay, LC-TOF, GCMS, TOF, HPLC, and GCMS, we have managed to identify about more than 1,000 compounds from the polygonominers or from kesum, but there is many not yet identified. Okay. So this is the ultimate aim of metabolomic study. You need to map into the pathway for the biosynthetic pathway. So this is the biosynthetic pathway that we are mapping with based on the KAGG database. So you can see that this is a terpenoid metabolism pathway for our own kesum. Okay, we don't need to use that model plant anymore. Okay, this is our own model plant, kesum plant. Okay, so we have come up with this mapping, okay, based on many compounds that we found in the terpenoid biosynthetic pathway. Okay, this is another plant, another pathway, flavonoid pathway. Also, we have found all these in our plant on the flavonoid biosynthetic pathway. Okay. So list of compound of interest. Okay, we have list a potential compound of interest to see what is the biological activity of this compound, and also to see if there is industry that will take up this later on. So this is the information that we find out based on the literature and some from our own discovery. Okay. So by using this, we now, after we have profiled all that compound of interest from our polygonominer, from the kesum, so we have come up with the biological activities. As I said, this is a metabolomic approach. Okay, it's a totally different from natural product approach, which is the from natural product. You have a targeted what type of biological activities that you want, then you study the plant. In our case, we study the plant. Then after that, we understand what is the biological activity of the plant. So we don't want to miss any information, very important information of the compound from the plant. Okay. So based on our study, we have found that the kesum plant produce highest anticholinesterase activities, okay, in their aqueous and also in the methanolic extract, okay. So this anticholinesterase normally been used for the cognitive study, okay, cognitive function study for the central nervous system, okay. Kesum also have this kind of property for the central nervous system study, so it can be used for the Alzheimer disease, dementia, and also for the Parkinson, because they are targeting the central nervous system. Okay. So the next, sorry, I cannot share with you on the Alzheimer work that we have working now because it's not yet published. Okay, but I will show to you what is our discovery for the another therapeutic compound. As mentioned, we have identified about more than 1,000 compounds, but see there is other unknown compound that is not yet identified. Okay, but luckily it's not that many. It's about like 80,000 unknown compound that not been yet identified from the kesum. So that's why this time we bring in, okay, the natural product expert. Okay, we have Professor Saidin from the Halu Olio University, and also we have Dr. Andi Rifki. He also working with me last year as my postdoc. Okay, so we have start this work last few years back with Prof Saidin. During that time, Andi is he's our master student. Okay, so we have managed to isolate a new novel compound. We call it polygonamines A. So the name is actually from the polygonaminous, okay, and then A is mean from the extract A, okay. So polygonaminous A. So we have tested this compound for the cytotoxicity activities, antioxidant activities, anticholinesterase activities, and also anti-HIV activities. Okay. Meanwhile, Dr. Rifki, he's working with the derivatives of this polygonamines A, which is he managed to isolate polygonamines B, C, D, and E. So we are working on we are still working on it on other study for the biological activity, or also on the anti-docking. To make sure that this paper can be published in very high impact journal, because it's something, to me, it's something breakthrough study that has been done by using the kesum compound. Okay, this work has been published in scientific report on the polygonamines A. It's a Q1 journals. Okay, it's not easy to get this published, the work actually, but Alhamdulillah, we managed to get it published with the help of our collaborator. In this work, we have collaborator from Chulalongkorn University as well. So they help us on the anti-HIV study. And so we have the as well we have the Prof Haydin, the one that come up with the new compound. Okay. So this is the isolation process. I will not detail on this. Okay, you can get this information based on my publication. Let me know if you want me to share with you my publication. I'm happy to share. Okay, just send me the email. Okay, so but I want to show here that polygonamines A produce the novel anti-cancer compounds. Okay, this is the first discovery of new novel compound from kesum to produce this anti-cancer compound. So there's no any study on that. That's why we managed to get this patented, Malaysia patent. Okay. So this is the anti-cancer properties. So it shows that polygonamines A can be used to treat the cancers such as the leukemia, colorectal, and breast cancer. This is been done based on the cell line study only, not yet on the rat study. Okay. But there is a study being done by my PhD student. She also using this polygonamines A, not polygonamines A, the extract of kesum to use as the protective effect. Okay, once the once the animal or the rat has been infected with the cancer, they give them the chemotherapy drugs, right? So use this kesum extract as a protective effect. So there is a very remarkable finding from the work. Okay. Okay. So from here, I would like to highlight here. Okay, there is a 6 compound isolated from polygonal means A. So one has been discovered but produced a novel compound. Okay, so we are still working on another 5 compound. Okay, so this is a molecular docking of the polygonal means A against the HIV protease. Okay, this also show very low binding affinity towards the peptidin A. Peptidin A is actually the protease inhibitor for the HIV. Okay.
Ryan, only 5 minutes.
Okay, sure. Okay. So I'll just go through very fast this one. Okay, this is some of the study on the insect repellent. So we have the extract of kesum. We put in that aquarium with the insect inside, and then after 1 hour, most of the insect already die. So we use this as insect repellent. So we have managed to come up with this product. We got that the trademark for this product, Alhamdulillah, and then we won some award for this product. Okay, so the take-home message for this work, actually, untargeted metabolomic approach reveal the potential of local herbs. In this study, we are talking about the kesum. Okay, metabolic discovery, discover a neurotherapeutic new compound. Okay, which is we are talking about the polygonal means A and other derivative, which is B, C, D, and E. And then metabolomic also reveal a potential biomarker for geographical population distribution. As I said that we can manipulate the production of this compound, lowland, highland, different temperatures, so can produce a lot of compound of interest. And metabolomic approach also can be used to characterize the valuable compound for the flavor and fragrance industry. The one that I mentioned that we know, we discovered that kesum have 3 notes. There's no other study has been done before. Okay. So this is some selected publication on kesum. You can let me know if you want to read on this publication, and please cite us in your paper later on. Okay, I would like to acknowledge people that working very hard with me for many years. Okay, Prof. Sahidin from Olio University, Dr. Anil Rifki, he has been with me from his master degree and until he's become my postdoctoral. So thank you a lot for all your help during the study. And other my PhD student, Dr. Azizan, Dr. Nurul, Puan Rafidah, she also working with me until now, working a lot on the biological activities. And then my PhD student working on the Alzheimer activity and other student. And also I would like to thanks the grant that we received from the MOSTI, Ministry of Science, also from Ministry of Higher Education and other UC grant. So this is the people that in UKM under my group, Metabolomics grant. So you can see the happy face working on the kesum. So we went for many exhibition. Okay, I would like just give me some minutes. Okay, I would like to promote our postgraduate program. So we have Master of Science in System Biology and also Doctor of Philosophy, PhD. Okay, please come to us if you like to do some PhD with us. Maybe you can send your docent here there in ITB. Maybe you can send your student to us for intern. So I received many from Chulalongkorn University, and also last time we received from the Korean University. So and then this is facility for metabolomics lab. So we have all the LC-TOF, the GCMS with the GC olfactory detector and so on. So this is transcriptomic, proteomic, transgenic, bioinformatic, and our greenhouse. So you can plant our plant here. And this is the research plot. So you can see very high, happy kesum there. So very big and very big kesum leaf. Okay, this is a mangosteen tree. Okay. So this is some analytical service that we provided. Okay, please contact us if you want to know detail on this. So there's the prices, but for the collaboration, we give you very special discount. Okay, so please collaborate with us. So also we have this analysis. Some of the researcher they don't know how to do the data analysis, right? So Dr. Arif will talk about that one later. But we also provide the service for the data analysis. You can send your sample for us to do the data analysis for you with some very reasonable amount of charges. Okay, just to show you some visit to ITB and IPB. So I met Prof. Al-Fahmi last time. Okay, thank you Prof. Al-Fahmi for inviting us last time. So just last but not least, we are organizing a third International Postgraduate Symposium, Biology, Biotechnology and Bioengineering 2021 in 27 and 28 October. Okay, so there is a speaker from the Netherlands University and also the Fort Warwick University. This is very, very young but talented researcher. Okay, I'm not that young, so I'm not, okay, but these two are very young. So please come and join us and see, because we have a session, a special session with the young researcher. So you can ask questions, you can ask their experience and so on. Maybe you can ask them if they have a place for you to be in their university in Netherlands and UK as well. So please register. All the postgraduate students are invited. So you can register as participant and also maybe as a presenter. We encourage you to register as presenter because it's only 5-minute presentation. So don't worry so much. Okay. So that's all from me. Thank you so much. Okay, thank you, Dr. Rifki. Back to you. So you can contact me here. Okay, if you have any questions and so on. Okay, I'm happy to share the slide. But please email me because I need to do some editing before I can give you the slide. Thank you. Okay, thank you, Dr.
Okay. Thank you, Dr. Ryan, for the wonderful presentation. Very wonderful. If you have any question regarding the topic, please write them down on the chat below. Next, we have our second speaker, my friend, Dr. Arif Izairi Zamani, also from Inbiosis. His expertise is in metabolomic data analysis using SIPCA, metabo analysis, XCMS, and identifying microorganism using Biolog. Well, before Dr. Arif present the topic, we have a video to watch. Operator, please.
Medica is one of the most dynamic and innovative companies within the Berka Group. The Berka Group consists of over 35 operating companies in over many industries, including technology, infrastructure, and engineering. Berka Medica has been in business for over 17 years and has established itself as the major player in the research and the chemical analysis industries. Its customers include many bodies in the government sector, as well as large corporate entities. And its industries include food research, agriculture, petroleum, chemical, environment, and clinical research. We believe that Berka Medica is very well suited to meet with all stakeholders to support the quality of life and to improve it in Indonesia. We fully support Berka Medica to meet with all stakeholders and form partnerships to improve the quality of life of people in Indonesia.
Okay. So that is a video from PTBNM. And now please welcome Dr. Arif with his topic overview of LCMS-based metabolomic workflow. Dr. Arif, the floor is yours.
Thank you, Dr. Andy, for your kind. Yep. And my slides are clear at your end. Yeah, I guess my slides are clear. So once again, thank you, Dr. Andy, for your kind introduction. Assalamualaikum and greeting everyone. I would like to express gratitude to the host, Nutritionalika Institute Technology Bandung, for inviting me to this great event. It's also been my privilege and honor to share this virtual platform with Dr. Niza and also especially with Prof. Ain, one of my supervisors during PhD study. So in this second session, I would like to share with you all about an overview of LCMS-based metabolomics workflow. So yeah, thank you to Prof. Ain for the great introduction on today's topic. I think it makes my task a bit easier, so I can skip that introduction on metabolomics part. So I begin with liquid chromatography mass spectrometry, or you could say LCMS, and these figures schematic of LCMS workflow for plant metabolomics provide an overview of LCMS-based metabolomics. So liquid chromatography mass spectrometry is a system that couples liquid chromatography with mass spectrometry. So liquid chromatography, as we know, the function is for metabolite separation and commonly use CAT column to operate at reverse phase, especially to retain a non-polar metabolite, as well as Healy column, which focus on the polar metabolites. And for the mass spec, we have three main components: ion source, mass analyzer, and detector. So the common mass spec, or perhaps we could see the current mass spec mostly in the tandem mode, which could provide the fragmentation of any molecule. So during the fragmentation, not only the parent ion could be detected, but as well as the daughter ion. So this will facilitate us to identify metabolite more accurately, not just relying on the mass accuracy of the parent ion itself. So we go to the next slide. So these are the example of technology in mass spectrometry. So here we could see some of the tandem mass spec. I mean the mass spec that could provide the fragmentation. Normally in a hybrid, for example, QTOF, quadruple time of flight, or linear triple quadruple with Orbitrap, and also quadruple with ion trap. And also there's a tandem of triple Q, triple quadruple, which are more reliable in order to run targeted analysis. Let's say we like to quantify metabolites. So the information that can be used not only at parent ion, but also at daughter ion. So this allows us to not to measure, but to identify and accurately measure the metabolite concentration in any samples. So for the QTOF, which have higher resolution, more accuracy, normally being used for the metabolomics, integrated metabolomics or metabolite profiling. So here we go to the next slide. So as being shared by Prof. Ain previously, to the metabolomics have two parts. So integrated metabolomics, or sometimes being known as a metabolite profiling, as well as targeted metabolomics, when we say metabolite quantification. So these are the examples of the metabolomic framework, which have been conducted on the microorganism, which employ both untargeted metabolite profiling and also targeted metabolite profiling, which focus on the organic acids. So this means that both framework can be conducted on same samples, and sometimes it might correlate each other. Let's say in this study, when the study tried to quantify the organic acid production from a microorganism, later the study tried to investigate the changes that cause different organic acid secretion. So that goes to the untargeted one, in which we like to investigate what metabolism being perturbed or under different conditions, which cause different secretion or expression of organic acids. So we go to the next slides. So type of sample for CMS based metabolomics. So besides microorganism and plants, also study done on fish, as shared by Prof. Ain. So more oriented either focusing on certain organs like spleen, or maybe as a whole body. There are also studies that focus on using clinical biological fluids, so normally known as metabolomics. And the study use either urine, most study use focus urine and also serums. So as we know, urine is we could say the waste metabolism from a host, which also contains some co-metabolites from any gut microorganism, whereas serums will contain some lipids. So combination information from urine and serum perhaps could give us some insight, or we could say as a metabolic signature on the host, and what a possible metabolic state of the host. So next we go to the example of sample extraction. As we all know, metabolomics are quite, we call it critical and heavily influenced by numerous factors compared to the proteomic and transcriptomic. So it is required for the, let's say, for the sample preparation like grinding to be in a fast manner and facilitate with liquid nitrogen to do the quenching process, as to freeze the metabolic state of the samples. And following that, the extraction, normally we use liquid extraction or solid liquid extraction. Some use Sohlet and even SPME. So for the experimental designs in doing metabolomics, this is quite a crucial, and a standard guideline have been proposed within the field on how to design the experiment in doing the metabolomics. So for the biological, there are several replicates, perhaps we could say, terms known as biological replicates, technical replicates, and as well as analytical replicates. So for biological replicates, the sample preparation are prepared from independent source. For example, we study on polygonal miners, so it's a species, but the preparation perhaps from a different source. Perhaps we study on the phenotypes, let's say pegaga, red pegaga or green pegaga, for example. So that each phenotypes, the source is different. So similar with microorganism, the culture of bacteria or the fungus. So it's from a different cultivation flux. So that's what we call it a biological replicate. So the indeed the preferred number of replicates, biological replicate, say more than 3, but it is highly advanced at the number of replicate, the size is 5. And for population study, not much standardized way. Perhaps there is a calculation normally known as power calculation, but most previous study, normally the sample size, they begin with n more than 300. So these are the examples of biological replicate. Let's say for the fungus species undergo different treatments, control versus treated. So for the each group, let's say control, we have 5 biological replicates and treated 5 different biological. And also for the population studies, let's say individual of within the age group or range with a diabetic diagnosis, for example, men versus women. So we have a population size of about 300. So that's for biological replicates. So what about technical replicates? So compared to the biological replicates, technical replicate involve independent performance of the complete analytical process. So as being discussed just now for the biological replicate, so it's from an independent source. Let's say mice from undergo same treatment, let's say treated with some natural product that we would like to investigate how it affect the mice. So each mouse considered as one biological replicate, so the total number, the size is 3. Whereas for technical replicates, so it's from the same source, from the one biological samples, but rather being processed or prepared in an independent way, means that got 3 technical replicate, but the biological replicate is considered 1. So it's just like one biological replicate with 3 technical replicate. So we go to the next slide. So another important point in experimental design, so the introduction of the quality control, or we could say a QC. So the quality control QC is a pool sample from all biological replicates in a given study, whether it's a treated or non-treated, whether it's a different phenotype, red versus green phenotypes or different ages. So how we prepare this pool QC? So we collect a small volume of each biological replicates, regardless what the treatments or what the groups are, and mix in homogeneous pool sample. So we prepare the replicate, and this pool sample, pool QC, which known as a QC, the importance is to highlight or to show the robustness of our system. In this case, the LCMS system. It has a same function with a technical replicate, which show how it consistent from each run to each run. Perhaps we say the reproducibility and also the robustness of the system. So as shown here in the figure, the PCA, you see the QC had been call it cluster in a group, like the robustness of SMS-based system that been used, which show no significant variation of due to the system itself. So that's for experimental designs. So when we run a sample and obtain data from LCMS, so one could expect the output in three forms. So first, it is a chromatography, which have intensity and also retention times, and also the mass spectra, list of mass spectra. So here we could say even at 0.9 minutes peak with intensity about almost to 10 to the power 5, number list of mass spectrum available. So this data could be projected in the 3 axes. So MZ value, intensity, and also retention time. So one could imagine this is from a sample, one sample, but how we gonna to compare from one group to another using this input? and even each group have, let's say, 5 biological replicates. So how we could compare 10 replicates or 10 samples using the input. Hence, we need to generate data metrics. So we need to transform all these 3 inputs, retention time, intensity, and MZ value into a data metric. So in preparation for statistics, this conversion is necessary. So the basic transformation of this SMS data input in data metrics commonly involve 3 steps. First, peak picking. Second, alignment across all the samples. So this is important for SMS system to be highly reproducible and robustness, as well as the last one, the algorithm to combine first the charge state, the isotopes, they got a dark, and also the ion clusters, and the available software for this data transformation in forming data metrics. According to vendor, there's a Porphyrosis under Brookers or Mesling under Waters. We have also open source that available, such as Metabolomic, SMS, MZMine, that can help us to transform the data input from SMS system into data metrics. And yep, we go to the next slide. So upon forming the data metrics, normally pre-treatment of the data need to be carried out. So first is normalization, is to reduce the systematic variation between each samples, specifically due to the system itself. So the common approach that have been conducted in biosis, so normally we spike internal standards such as gallic acid, gallic acid, riboflavin, etc. in all samples, regardless which group, either treated, non-treated, we spike in all samples. So here we could see the figure that shown here, the consistency of a peak that represent from a spike of gallic acid in all samples. And this is another point that highlight the robustness of the SMS, which is important, and later be used to normalize samples, because as you could see, there different samples have different spectrum, different chromatography, as you could see here, but the spike sample are needed to normalize all these samples to eliminate the possibility variation due to the system itself. So another way, besides using internal standard that spike into all samples, we use a relative abundance, which is also commonly be used by adjusting the ion intensity based on their ratio to the internal standards. Another approach, like normalized to constant sum, normalized to reference samples, and also sample-specific normalization, either using dry weight or tissue volume. In data pre-processing, besides normalization, most people like to scale their data metrics. So the common one is auto-scaling, rescaling, and Pareto, which have different goals. So it depends the needs of the research itself, how to structurize the data. So either to focus on answering, correlating with the phenotype, or would like to consider all metabolites in the sample have a same impact. So it depends on the needs of the research or the study. So yeah, we go to the next slide. So as we discussed just now, there are several software that are available online that could help us to provide this not only data transformation, but also pre-processing, including normalization, scaling, and as well as transformation. So among them is Metabolomic, MZMine, MSDAL, SMS, and etc. So the common data input, perhaps you could see the universal one, commonly accepted in most open source software like MZSML or NetCDF. So these are the common data input that accepted by open source, meaning that if we running our sample, regardless using a different vendor, either Waters, perhaps if you like to use this open source, perhaps we need to transform or need to provide the data in this kind of format. So we go to the next slides. Here I would like to share you a bit detail on one of the popular open source software for the metabolomics known as Metabolomic. Here's the link. So it's a popular choice together with SCMS, Syncapi, MZMinds. And the first version of Metabolomics was focusing only data normalization and statistical analysis, comprising univariate and also multivariates. Later, they start to develop, and I think now it's up to version 5, if I'm not mistaken. So the current metabolomics could support functional analysis for targeted metabolomics, not only to have a raw spectra as an input and to transform it to data metric, but as well as for functional analysis for targeted metabolomics, and also integrated with other omics such as transcriptomic, genomic, for the interpretation of global metabolomics data. So once we have the data metrics and we already pre-process the data metrics, so most metabolomics works, they do it in a comparative manner, meaning that there is let's say there is a pair group, a pair samples, for example, wild type versus targeted mutagenesis, or one phenotype to another, red versus green, or different ages. So indeed, metabolomics could be performed as like profiling, but when we do it in a comparative way, it gives us more insight what possible changes happen in the cells when undergo certain perturbation or certain factors. So normally for comparative metabolic analysis, univariates and multivariate analysis are being used. So for univariates, perhaps we could say statistical analysis are commonly used, similar with other kind of arithmetic designs. So among them are like pair t-test, fold chain analysis, also even ANOVA. So the tests are used to find the significantly different metabolites in both unpaired or paired study in a comparative manner. And p-value are commonly used for assessments coupled with a false discovery rate. And for multivariate analysis, so in contrast to univariate, the analysis consider the combinatorial effect of the multiple variables. So it could be categorized into two, either unsupervised and supervised technique. So for the unsupervised, the most popular choice are PCA, principal component analysis, as well as SOM, self-organizing map. And for the supervised, the method use label information to construct a model. And one of the popular ones is partial least square discriminant analysis, also random forest, as well as support vector machines. Normally for the supervised analysis, for supervised approach in multivariate analysis, normally related to the model building. So cross validation and permutation tests need to be carried out to ensure the genericability of the constructed models.
Dr. Arif,
Yep.
only 3 minutes.
Okay. Thanks, Dr. Andi. So here I share a bit on some of the work from Metabolomic group, let's say on the principal component analysis. So this study investigate between wild type and mutant. So here we could see the wild type and mutant being clustered into two, as explained at the first species. So similar with another study conducted from Metabolomic group on the Weigela tribolata leaf. So you could see the separation between soil and leaf litter in the first species. So here what we could explain, or what perhaps what the input we could get. So we could say approximate about 67 of metabolite, detected metabolite, contribute or involved in the differences between soil and leaf litter, whereas another 20% perhaps could involved in explaining between soil litter with a leaf. So this how PCA being used. So it's try to localize, to group the sample. What is the common ground between them? So another one is the supervised one, PLSDA. So normally being employed to discriminate, to find the metabolite that cause the differences between group. For example, another study from metabolomic groups to which investigate the differences between control and treated. So here from the PSDA, we could determine or pinpoint which metabolite, as indicated in the red, that cause significant difference between the control and compared to the treated one. So these are the examples of the approach, multivariate analysis, which help us not to identify significant metabolite, but as well to find biomarkers in studies. So there's also a study that employ OPLSDA and try to correlate what metabolite being upregulated at one end and what metabolite being downregulated in another end. So once we knew the list of MZ value that show that changes between a study group, normally we like to know what are these metabolites. So the identification is crucial and so necessary. So there are several online database that commonly being used in metabolomics for metabolite identification, which include Medlin, which cater most analysis using standard using QTOF. Also got mass bank, we cover different kind of instrument, including QTOF, ion trap, and etc. And also we have an in silico calculation from the MedFrag, which the metabolite could not be identified. Perhaps in silico calculation, which require not only information at parent ion, but also daughter ions. So bear in mind, as been shared by the first speaker, that not all metabolites could be annotated, as being shown here. So there are many unknown metabolites that are yet to be discovered. And as with the improvement of the mass spec, with the increase of high sensitivity, more unknown metabolites will be explored, especially in our region, Southeast Asia, which live with biodiversity. So many exciting high prospect compound could be yet to be known. So there are different level of metabolite identification. I will share you the slide, the formats. So it's according to the metabolite initiative, standard initiative. And yeah, for the targeted metabolomics, as been shared in the first slide. So it's more direct, just to quantify metabolite, in which the output we know the expression, the level of metabolite from one sample from one group to another. So first, the basic principle is first we need to have a standard, the target the metabolites, and try to optimize the best approach to have the good spectrum, and later to develop standard curve. So normally in analytical journal, they also require the limit of detection and limit of quantification for this type of analysis. So as be shared by the first speaker, the goal, perhaps the ultimate aim in metabolomics is to have a metabolic network, a pathway, a part of system biology. So when we compare with other omics, perhaps the could give us a more significant story inside what actually happened, not only at metabolomic or also phenotype level, but also at the proteomic, transcriptomic level, how the expression correlate with the changes at metabolite levels. So these are the example of the network that been visualized using the metabolomics input along with targeted enzymatic assay. So the diagram could be drawn either using Vanted or even in a metabolic analysis. Also, there's a study not using any software, but rather try to visualize the pathway. And for example, this one, which try to differentiate between control and activation, how the expression differences and how it happened in a network, in the pathway. So it gives us some insight overview what could actually happen. So the take-home message, I'm sorry for taking a bit time. So as being discussed just now, metabolomics, they have two main parts, untargeted and targeted metabolomics. And the experimental design is crucial, is important. Number of biological replicate as well as technical replicate, and also QC and data pre-processing using either vendor software or available online database, multivariate analysis in order to find biomarkers, metabolic identification, as well as metabolic network. So I would like to say thank you first to the host for inviting me to this such great event, to the Metabolomic group from Embraces. Most of the work be shared, study be shared is from the Metabolomic groups. I would like to extend the acknowledgement to Embraces, research fellow, officers, lab members, especially my PhD supervisor. Thank you. Yeah, back to you, Dr. Andy.
Okay, thank you very much, Dr. Arif, for the insightful presentation. Very technical and a lot of help for us. If you have any questions for topic from Dr. Arif, please write down the chat room. Okay, so our last speaker, Dr. Nizar Hafiana. Okay, Dr. Nizar Hafiana, he is a lecturer at Organic Chemistry Division, ITB. And right now he focused on NMR metabolomics, proteomics, transcriptomics, and genomics or genomics. And also he is expert in NMR elucidation structure. Let's welcome Dr. Nizar with this topic today about proton NMR based metabolomics. Dr. Nizar, the floor is yours.
Okay, thank you very much. Good morning and Assalamualaikum warahmatullahi wabarakatuh, everyone.
Waalaikumsalam warahmatullahi wabarakatuh.
First, I would like to thank to Dr. Andi for your kind introduction, and also to Pak El Fahmi and Pak Agus from Nutracetica for inviting me to this webinar. It is a big pleasure for me to be here in this webinar and in the same stage with Professor Ain and Dr. Arif. I think I have met with Professor Ain 5 years ago when she visited chemistry department. So we meet here in online. Okay, at this moment, I would like to present about proton NMR-based metabolomic with the case luwak kopi. Okay, so for the introduction, I want to repeat again the definition of metabolomics. So metabolomics consists of two words, namely metabolome and omics. So metabolome can be defined as the complete collection of small metabolite in cell, organ, tissue, or organism. So the key point here in metabolome is the complete collection of small metabolite. And then the second word is omics. So omics can be defined as the field of life sciences that aims to identify, characterize, and measure all the biological molecules involved in the structure, function, and dynamics of cell, tissue, or organism. So the focus here is identify, characterize, and measure all the biological molecules. Therefore, we can define metabolomics as the field of life sciences applying high throughput technology to identify, characterize, and measure all the small molecule or metabolite in given biological sample, including cell, tissue, or organism. There are some important points in metabolomics that we must understand carefully. First, metabolomics is multidisciplinary science. So at least it involves chemistry, biology, informatics, and statistics. Second, metabolomics use comparison method for obtaining the information what we want. For example, if we want to find the effect of, for example, effect of the elicitation in the plant tissue culture, what metabolomics do is compare the metabolite profile between the treatment with the control. From that, we can extract the differences and also the similarity between them. So from that, we can know the effect of the elicitation. So that is the basic principle of metabolomics. And then metabolomics work with extract or fraction. So it has some advantages. First, the preparation is simple, especially when we work with NMR-based metabolomics. And second, we don't need to separate the compound. However, it also has disadvantage. First, for example, for metabolite identification, you can imagine for determine the structure of your compound is difficult. So here we have to identify metabolite in the crude extract containing 1,000 probably compound. So it's very difficult. Okay, and then when we perform metabolomic experiment, we have to do it with consistency and uniform from beginning, from the sample collection, sample preparation, measurement, data processing, and analyzing. So we have to do it with consistency. And the last is database. So metabolomics need database. The database is used for metabolic identification, and then for interpreting the result, and also for metabolic pathway mapping. So we need database. This is an example popular database, for example, HMDB, MMCD, and BMRD. Okay, NMR has some advantages. First, it is non-destructive analysis. That's mean the sample that have been used for NMR measurement can be used for another analysis. And then NMR provide information of molecular structure comprehensively, including stereochemistry. So it is very important for chemists. And then the preparation of NMR-based metabolomic is very simple and fast and easier. And then the measurement relatively is very fast. For the sample of metabolomic, probably it need around 4 or 5 minutes per sample. And then for the quantitative analysis, NMR doesn't require calibration curve, and the result is very acceptable. And the last and the most important is the NMR has high reproducibility. That means when we repeat again the experiment with the same method, with the same parameter, the result will be same, more or less. However, NMR also has disadvantage. First, it's less sensitive if we compare with the previous presentation, LCMS or GCMS. NMR is less sensitive. Second, proton NMR spectrum contain overlap signal, so it is very difficult to identify. And then NMR interpretation is difficult because we cannot compare with the data piles in the NMR, for example, in the instrument. So it's very difficult. And actually, there is the last. The last is expensive, especially in Indonesia, because the maintenance is very expensive. Okay, and then however, if we compare with another metabolomic instrument, we can see here NMR-based metabolomics superior in some category. For example, in sample preparation, reproducibility, and quantification. And yeah, it's poor in it's poor in identity and number of metabolite and sensitivity. Okay, so this is the flow chart of NMR metabolomic experiment. So the sample can be organism like plant or bacteria, fungi, and then it also can be a herbal product like jamu or plant biotechnology object like plant culture. So the sample can be directly extracted, or it can be administered to animal or human, and then we take the urine, blood, or other biofluids and tissue, and then we perform extraction. For extraction, we can use directly the NMR solvent or normal solvent. And then we perform measurement, producing spectra, in this case proton NMR spectra, and then we perform data processing. In this data processing, is already mentioned previously by Dr. Arif. It consists of, maybe the difference is here, bucketing, and then alignment and normalization. Okay, and then after we process the data is ready for chemometric analysis. Normally can be performed with commercial software like Simca, Unscramble, MATLAB, or pre-open software like Orange or the previous one, Metabolomic, that's explained by Dr. Arif. And then we perform metabolic identification, and the last, we interpret the result. So this is normally the flow chart of NMR metabolomic experiment. It's a little bit similar. It's similar, more or less, with LCMS based metabolomic experiment. However, the difference is maybe in the beginning here, and also for the identification. Okay, for example, we have perform sample collection. The next step is extraction. The extraction method should consistent and for sure reproducible. And then the selection of solvent should be based on the physicochemical properties of targeted metabolite. So you know there is no single solvent can extract all metabolite. For example, if we work with genomic or proteomic, yeah, most of the gene or proteins can be extracted. However, for metabolomic, it's very difficult to extract all metabolite because the polarity is vary. So it's very difficult. So we have to know our target metabolite, at least the polarity of our targeted metabolite. Based on the solvent type, there are two types of extraction for NMR-based metabolomic. First one is indirect extraction. In this technique, we use a regular solvent like normal methanol or water or N-hexane or chloroform. Because we use a regular solvent, we can obtain more abundant metabolites. For example, we don't have to limit the volume of the solvent because it's cheap. So we can obtain more abundant metabolites. So the solvent is dry, for example, with a rotary evaporator. So we get the dry extract, and then the dry extract dissolve in the diuretic solvent. So if we see here, maybe the step for the extraction is too long if we compare with direct extraction. So in the direct extraction, we use the diuretic solvent or NMR solvent. So the solvent can be directly transferred into NMR tube without evaporation process. So that's make the preparation is simpler, easier compared to indirect extraction. However, because if we see the price of the NMR solvent is expensive, so normally people perform the extraction using NMR solvent only 2 milliliter or 5 milliliter. So because you can imagine the price is expensive for Indonesian. Yeah, okay. And then because the volume is limited, so the quantity of extract metabolite is also limited if we compare with indirect extraction. So for example, here the procedure of coffee extraction using direct extraction. This procedure is very simple. First, we have the sample around 200 milligram. We put in the plastic tube and then mix with 1 milliliter B2O containing TSP. And then the sample was sonicated for 20 minutes and then incubated at 90 degrees Celsius for 30 minutes. And then the sample was still on the water for 10 minutes and then centrifuged at 5,000 RPM for 5 minutes. And then we take around 400 microliter of this pernatan and mix with 100 microliter of phosphate vapor. And then the sample is ready for NMR measurement. So you can see here, the procedure is very simple, easier, and fast. More or less for this procedure, it will take time around 2 hours. And then we're ready for NMR measurement. Okay, so before NMR measurement, we have to make sure that the volume of all samples must be same. Also, we have to use the same NMR tube type for all samples. And then when we perform NMR measurement, we have consistent with the parameter for all samples. So this is an example of the NMR parameter measurement. For example, the acquisition time is 2.72 seconds, and then the relaxation delay is 2 seconds. Number of data point around 64,000. Spectral wide is around 8,000 hertz, and then the number of scan is around 128. So this is an example of NMR measurement parameter. Okay, next. So after we obtain the spectra, we perform data processing and chemometry. So in the data processing, first we have to perform spectrum quality check. We have to check the quality of the proton NMR spectrum. For example, here the green one is the bad spectrum because it has low resolution, So that means we have to mean to refit the measurement to yield a good spectrum like the blue color. Yeah. Okay, like this one. And then we perform data extraction. So data extraction consists of alignment, bucketing, and normalization. Probably the difference when we do data extraction between NMR and LCMS or GCMS is here. So in the alignment, we put all spectra in one window and treat it as a group. And then in bucketing, we divide the spectra are divided into same wide, yielding 100 or 1,000 data of bucket. So the last is normalization. I think normalization already explained by Dr. Arif. Normalization normally commonly used first using the reference or internal standard, and then the second is with the sum method. And after that, we have the ready data set. We perform chemometric analysis or data reduction with statistical analysis software. There are two types of data reduction. First one is unsupervised, and second is supervised approach. I think it's already explained clearly by Dr. Arif. Okay, for example, this one is the score plot. So the score plot reveal the similarity and differences within the sample. So if the sample having high similarity, they will close each other. For example, here, so if we see from the first component here, the sample honey here and natural is close each other. That means the profile, metabolic profile, is more or less similar or has higher similarity compared to the weight.
Dr. Nizar, only 3 minutes left.
Okay, so, okay, I will do it fast. Okay, and this is the loading plot. Okay, the next is metabolic identification. This is the most challenging step. Yeah, you can imagine we have to identify the metabolite in the fruit extract of proton NMR spectrum. Okay, there is some step. First one is we have to detect the fingerprint signal. After that, we can validate the fingerprint signal with 2D NMR technique, including JDSOP, COSI, TOXI, SMBC, and SQC. And then we also can compare the spectrum with reference NMR spectra. And then we also can verify the result with the data in the literature and database. Okay, this is an example how we can apply NMR metabolomic in some research. This is the easy one, namely the comparison study. I think comparison study is the basic metabolomic study. So we compare the metabolic profile. For example, different species, and then different variety or cultivar, or different organ, or different times, or different geographic origin. Okay, so this is Lua copy. So I will demonstrate briefly how NMR-based metabolomic apply for studying Lua copy. So Lua copy, I believe everyone here already know, is the most expensive coffee in the world. So the price for 1 cup of coffee is around $4 until $100. and then the wild luwak coffee is more expensive than the caged luwak coffee. Why? Because the taste of luwak coffee is unique, and then the supply is very low, and then for producing luwak coffee require extensive labor. So that's the reason why luwak coffee is expensive. However, the study on chemical profile of luwak coffee is still limited in the literature, and also there is no work reporting the anti-diabetic activity of the coffee beans after fermented by dissipated or the luwak. Okay, so here is the procedure, experiment procedure per luwak coffee study. So here we use 3 types of green bean. First one is the white luwak coffee, and then the second is caged luwak coffee, and the last is the regular coffee as the control. So we ground the green bean into powder, and then we extract it with B2O, and then we perform measurement, and then we perform NMR data processing, and then we perform component identification and chemometric analysis. So producing chemical profile of luwak coffee. And beside that, we also do extraction using normal solvent. And then we perform two assays, namely antioxidant assay and alpha glucosidase inhibitory activity assay. From that, we will know the anti-diabetic activity. So by combining this information, we hope we can find the bioactive marker. So actually, this paper is already accepted in food chemistry this year. Okay, so this is the spectrum of proton NMR of luwak coffee. So here we can see that we can identify around 17 compounds. And if we see here, the signal belong to caffeine, sucrose, trigonelline, and chlorogenic acid were clearly detected in the spectra. That means these compounds are major compounds. And besides that, we also can identify another acidic compound like lactic acid, quinic acid, acetic acid, malic acid, citric acid. And also we can identify 3 amino acids, namely alanine, here, asparagine, and GABA. So this identified compound were further confirmed by analysis to the NMR spectra, including GRISOP, COSI, and TOXI. Okay, so this is the score plot, the result of chemometric analysis. So here we can see in the first component, we can separate between the regular coffee with the luwak coffee. And then in the second component, we see here the caged luwak coffee is separated from white luwak coffee. And then for identifying the metabolite responsible for this characterization, we investigate the loading plot, the corresponding loading plot. So here, for example, we can see caged luwak coffee is characterized with lactic acid, while white luwak coffee is characterized with malic acid and citric acid. And the regular coffee is characterized with sucrose, chlorogenic acid, and caffeine. Okay, so this is the result of alpha glucosidase inhibitory activity assay. So here, the important thing is the white luwak coffee had the highest value of the activity. And then here, according to statistical analysis of Pearson correlation coefficient, we find that malic acid had a positive correlation with alpha glucosidase inhibitory activity of the coffee. And then this is the result of antioxidant assay. So the result more or less similar with the previous one. So while luwak coffee had the best antioxidant activity, and also the malic acid had the positive correlation with antioxidant activity of the coffee. Okay, so this is the conclusion for luwak coffee experiment. So luwak coffee had a unique metabolome, with characterized with higher concentration of malic acid, trigonelline, citric acid, and lactic acid, and also characterized with lower content of caffeine, sucrose, and chlorogenic acid. And we find here that uwa coffee has a better potential as functional food than the regular coffee, because it has the anti-diabetic activity is better than the regular one. Okay, so this is other NMR based metabolomic topic in coffee research that we are doing now. So you can read it. Okay, so at this moment, I would like to thanks to first to Menristek Dikti and ITB for the funding of this research, and then from farmer group, kelompok Tani Rahayu, and Koperasi Produsen Makanan Tani from Gunung Malabar and Gunung Manglayang. Okay, and this is my team is very small, so this is, there is Propiana, Prof. Ois, and some student and me, and also my colleague Elvira. Okay, thank you very much. I give you back to Dr. Andi.
Okay, thank you very much for all speakers. Next, we move to the Q&A session. I think we have any questions here for every speakers. Okay, maybe we start from a question for Dr. Ain. Okay, Dr. Ain, maybe you can see on the chat, but I can read for you. From Arya Noor, how do you determine the terpenoid biosynthesis pathway in polygenome? Do you compare with another plants? And the second question, do you discover something new or unique in that biosynthetic pathway? Please, Dr. Ain.
Okay, thank you for the question. Okay, so for the biosynthetic pathway of terpenoid, so basically we based on the KEGG pathway that available in the database KEGG. So what we did is we mapped the compound of terpenoid that we found from the study, and then we mapped into the KEGG map pathway. So from there we come up with our own terpenoid biosynthetic pathway for the polygenome minus. So there is a unique actually in our kesome biosynthetic pathway. So we not yet published the work. So I'm still working on the draft because it's not that easy to convince the international people, that we found some interesting pathway in our plant. So I'm still working on the draft of that. Yeah, thank you. Dr. Andi, you're mute.
Thank you, Dr. Ain. Maybe the second and the third questions, quite similarity from Dr. Yossi, UMT, and Dr. Hagar from ITB. How did you correlate the metabolites of plants with biological activity through metabolomic approach? And then from Dr. Hagar, regarding your approach in polygenome means isolation, was it based on bioassay guided isolation, or did you find it from the metabolomic approach? Okay.
Okay. Thank you for the question. So I answer the first one. So the question on how to correlate the metabolites plant with the biological activity. Okay. So basically, as I mentioned, we did the profiling first. We just want to know what type of compound that produced in our kesome based on different treatment given, the temperature treatment given, and also on the location given. Okay, so from there, after we have done on that, so we found that there is an interesting compound there based on the literature that also have found in our plant. So we have come up with that biological activity based on the information available on the literature, and then it shows that our plant produce a very good activities of the different anticholinesterase, and as I mentioned on the anti-cancer and so on. So from there we found that this plant have a lot of biological activities. Okay, the second questions on the bio-guided or not bio-guided. Okay, so basically to get the isolate of this new compound, we based on the metabolomic approach, which is we found that there is a lot of unknown compound that cannot be identified by using the metabolomics approach. So we are assuming that this must be a new compound. Okay, so from there, Pak Saidin have done on the isolation and so on. While doing the process, he's not doing the guided, the normal guided experiment, which is every fraction you test on the anti-microbe, you test on the antioxidant and so on. He's not doing that. So he just isolate the compound until it's pure enough to be the new compound, because that's my target at that time. I want to have a new compound. So not the ordinary compound, the known compound. I want a new compound. So he managed to get the new compound, and then this new compound is meaningless if you not doing if this compound doesn't have any biological activity, right? So that's why we have tested these biological activities by using different approach. So we test with the anticholinesterase, for the antioxidants. We test on the anti-cancer and also on the anti-HIV. So actually the anti-HIV one is the we found that there is a paper that I share in my slide on the Venico side. This compound produce anti-HIV activity. That's why we test on our compound as well. So it shows that ours is better than them. That's why we managed to get that one published in the scientific report. Okay, I think I answered the question. Thank you.
Okay, thank you very much, Brian. Good answer. And then this is from Salina, Dr. Arif. When do you spike your internal standard? When do you spike your internal standard? At the beginning sample processing or after sample processing, then spike internal before sample analysis? Please, Dr. Arif.
Thank you, Salina, for the question. So, but the normal routines normally that we conduct, we spike the samples. We spike the internal standard in the samples prior the analysis of SMS. So meaning that once we finish preparing samples, so we spike the internal standard, let's say synthetic compound or whatsoever, in all samples. Then we put it in a vial, and then we analyze using SMS. Yeah.
Okay, thank you. The second question from Dr. Hega. How can we consider when to use this supervised on the unsupervised method for analysis? Please, Dr. Arif.
Sorry, Rutani, come again. In which question?
From Dr. Hega. How can we consider when to use the supervised or the unsupervised method of analysis?
Okay, so I thank you, Dr. Hega, for the questions. Normally in metabolomics, yes, there is supervised and non-supervised analysis. First we begin with non-supervised, in which we like to observe how the sample group by themselves, is it correspond to our experimental design or towards our hypothesis. Let's say we are investigating let's say treated and non-treated samples. So is it the sample behave according to what we had conduct on them? I mean a group for treated and non-treated. So for the supervised is a follow-up. Let's say we like indeed the sample being clustered into two. So we like to know which metabolite that cause the differences between one group to another. So then we conduct supervised, either using discriminant analysis or any other available approach. Yeah, thank you.
Okay, thank you. And I think questions from Wisdawati. How about if I want to conduct metabolomic study using same species, for example, curcuma longa from some different regions in Indonesia, and I take 3 replication from each sample, so I will have 15 data to analyze using SIMCA. Is it okay with just have 15 data? Maybe you can give the strategy. Thank you, Doctor. Yes, please.
Thank you for the question, Mr. Wati. Actually, it's allowed to have the minimum of biological replicates about 3, but could say it's a current trend or requirement in most publication to require us to offer to provide more biological replicate, let's say up to 5 biological replicate or more, as to show that the consistency of our sample that be used in the experiments. And perhaps we could not include the use of technical replicate, but certain study they use pool samples to address the variation due to technical. But it's suggested to have 5 biological replicate from each location. That's my humble suggestion. Yeah, thank you.
Okay, thank you very much, Dr. Arif. And then we move to another question for Dr. Nizar. How to apply of metabolomic in food chemistry or food science? This question is from Nova Kurnia. Please, Dr. Nizar.
Okay, thank you very much for the question. So if you want to apply NMR-based metabolomic in food science, so I think we must start with the easier, namely with comparison study. So, for example, if we have, banana, so there are many types of banana. Maybe in Indonesia is Raja Mas or Ambon and so on. So actually you can compare the metabolic profile between the different type of banana. So that is the simple one. We start with the simple one because if you want to go further, it's a little bit more sophisticated. So it's a little bit difficult. So we can start with the my suggestion is first we can start with comparison studies, and then the second we can start with our local food or our local food. For example, like oncom, maybe for Sundanese. So actually there is no work about oncom in metabolomic, I think. So we can, for example, we can monitor the time of the fermentation process of oncom from the beginning until the end, how the metabolic profile of oncom change during the fermentation, for example, like that. So I think for applying the metabolomic, we can start with the easy one, and the object is around us. So maybe that is the answer. I hope it can answer the question.
Okay, thank you. The second question for you. Some software includes DOE application. I don't know what the DOE. Could you explain about your application, how we can optimize that in the future?
Sorry, can you repeat it again?
From Rizwah Yuli. Some software include DOE application. Could you explain about DOE application, how we can optimize it for the future? I don't know what DOE.
Me too. From who?
Rizwah Yuli. Where is this Wahyuli? Below Immanuel Dani. You can see on the chat.
So, okay, I think I will answer it in common. Okay, so for NMR based metabolomic, we use statistical software, okay, like the open access software, like Metabolomic, and also the commercial one like Unscramble or Simca. Actually, I use Simca here. So the or MATLAB. So for actually in the multivariate data analysis, so we, for example, here we in NMR based metabolomic we perform a bucketing. Bucketing is we divided the spectra become small part or small bucket. So for example, if we have if we do bucketing from 0 ppm until 10 ppm or proton NMR spectrum, and then the width is around, for example, 0.04. So more or less we will get around 25,000 buckets. So there are a lot of buckets here. So what this bucket become our data. So become our input data. So and then what we do, we process it using a software. For example, the software can be processing NMR software like Mestrepnopa, or in this I use SCD Lab, and then we do bucketing, and then we do normalization. After that, the data is ready for multivariate data analysis. So in multivariate data analysis, for example, you can imagine previously I have 25,000 buckets, and then multiply with the number of sample. For example, the number of sample just 4, for example. This is only example. So in total, we will have around 10,000 and 100,000 data. So it's a lot of data. So with a multivariate data analysis software, we can reduce the complexity of the data, become small component. So we call it like, for example, for PCA, we call it as a principal component. So principal component actually the representative of major data. So probably that we do with the software. I hope that can answer the question.
Okay, thank you. I think from Ari, we have 2 questions for you. First question: how to separate the overlap signal in NMR data? What is the purpose of picking or bucketing in data extraction? And then it is a compulsory step in untargeted metabolomic approach. Please, Dr. Nizar.
Okay. First one, I forget for the first one. What is the first one question?
How to separate the overlap signal?
The overlap signal. Okay, the overlap signal is difficult. Maybe it can be solved with deconvolution. So the convolution is like we spread the signal. Okay, so maybe we can model it. So this is, for example, we have like 3 or 4 peaks. Actually, this is the overlap signal. So we can do it using the software. The software will model it and try to figure out what is the signal is. So like that, or we also can do picking manually. can do picking manually. We take the certain picks one by one like that. Okay, and then the second question, I think I already answered it in the previous one. So we do bucketing for obtaining the data for chemometric analysis. So in bucketing, for example, here I will show you. May I show the slide, Dr. Andi?
Yes.
Okay. For example, okay. Okay, can you see my slide?
Yes.
Okay, so first in the data extraction, we perform alignment. So alignment, we put together all the spectra in one window, and we treat it as one group. Okay, and then we do bucketing. Bucketing is like we cut the spectra, okay, like this. So we cut the spectra, for example, from 0 ppm until 10 ppm. So we cut the region here. So from each bucket, we will get the value of the integration for each bucket. So and this data become our input, become input for the chemometric analysis. And then for the untargeted yeah, we have to do bucketing for untargeted. Normally for targeted analysis, we can use like a bucket, like pick picking. So pick picking is normally we just take the integration of certain picks, but bucketing is like we divide the spectra, become certain bucket. Okay, maybe that's the answer.
Okay, thank you. Another question for you from Nancy Juliana. As we have two questions. Any recommendation for NMR database? She need free access or pay for plant secondary metabolite. And then the second question, do you have NMR for metabolomic in ITB?
Okay, the first question is from my senior actually. Thank you for the question, Ibu Nancy. Okay, the first question. Sorry, I forget the question. For the first one is
the first one. Any recommendation for NMR database?
Oh, NMR database. Okay.
Yes.
Normally, what I do, so I take the reference spectra from several database like HMDB and then BMRB. However, if we work with metabolic secondary, it's very difficult to find the database for metabolic secondary, especially if the metabolic secondary is very specific for certain plant. However, if we want to get the common metabolic spectra, for example, like caffeine or apigenin and common flavonoid, we can find easily in the spiral database like BMRB. However, if you want to get the specific metabolic secondary, it's very difficult. It's very difficult to find. That is the challenging step for NMR-based metabolomic. And then in ITB, actually we also accept the sample for metabolomics. Maybe we can talk later with Ibu Nancy for talking technically. maybe that's the answer.
Okay, thank you.
Thank you.
I think we will answer 3 questions for I think this is for Dr. Ain from Ms. Marcia Stefania. What is the best analysis tool to identify a certain bioactive compound from microbial extract? We only know that the compound's activity has protease inhibitors, but we have no profile of control to compare it. Is Dr. Ain?
Oh, okay. I cannot find the questions, but it's on the microbial disease, right? Okay, so for the microbial metabolomics, there is a different way of extracting the microbial. So you need to do some, we call it quenching techniques. Okay, quenching techniques. I think maybe Dr. Arif have shared on his presentation on the quenching technique. So you need to do this first, okay? And then microbial normally they are not volatile compound, I can say that. So you can use the derivatization techniques, okay? If you are using the GCMS, but for the LCMS, you can just extract just a normal extraction using different extraction technique. But what I can suggest is that for the microbial extraction, normally we get more polar compound than non-polar compound. So because there is a lot of primary metabolites there, then also the secondary metabolite, normally they go for like antibiotic or some sort of compound. So normally it's go on the best methods by using the methanol extract or maybe the ethanol extracts. Okay. So I hope that I answered the questions.
Okay, thank you, Dr. Ain. Okay, the last question to answer, I think to Dr. Arif from, I think Mr. Sinurat, right? I want to ask you about the data result of the experiment using Masling. I difficult to interpret and match structure. Our sample is from varied seaweed. May you have a suggestion to solve it? Thank you.
Thank you, Dr. Sinurat, for the question. I presume so the sample is switched from many environments, so perhaps it could have a high salts content, and perhaps it could contribute to the high numbers of metabolite with a dark. So perhaps my suggestion, perhaps we could export the spectrum, try to analyze with open source software. Let's say we go with SAMS or MZMine. Perhaps it could help us to properly identify or to say picking to allow us to better identification. I think that's my humble suggestion. Yeah.
Okay, thank you. I think that's all for the session of Q&A. Next, we move to photo session. But before that, I try to give summary for you, for every speakers. From Dr. Ain, untargeted metabolomic approach reveals the potential of local herbs. Second, metabolomic discover therapeutic new compounds. Metabolomic reveal potential biomarker for geographical and population distribution. Metabolomic approach characterize valuable compounds for flavor and fragrance industry. From Dr. Arif, start from sample preparation, how prepare the data, and then analyze it. So the output of the study is we will able to identify biomarker, for example, pegagan, that could have diabetic patients. So the metabolomic can be used to identify the metabolites. In addition, we could have an idea what metabolic change happening in the study cell by connecting between identified metabolites to form and pathways. And the last from Dr. Niza, Luwak coffees had a unique metabolome. Luwak coffee were characterized with higher concentration of malic acid and so on. Luwak coffee had better antioxidant and alpha glucosidase inhibitor activities. Luwak coffee possess a better potential as a functional food than the regular coffee. Ladies and gentlemen, I would like say thanks for you and to all speakers for sharing their valuable knowledge. Now we will leave this out to MC for the official closing. Please, Mrs. Nurin.
Okay, thank you, Mr. Andi. So that's all for today's session. Next, we will have a photo session for panelists and hosts. So please, for panelists and all the speakers, please turn on your video. And okay. And then for operator, please stand by. Okay, operator ready?
Ready.
Okay, so I will count in 3, 2, 1, smile. Okay, maybe once again. Once again, yeah. Okay, I will count again. From 3, 2, 1, smile.
Okay.
Okay. Okay, thank you, operator. And for participants, I would like to remind you once again to fill the questionnaire and the attendance on the given link to claim your certificate. Now, before we end this webinar, I would like to show you one of our program of this year, which is NMR intensive class. And here is the preview of our activity. For the operator, please show the video of the NMR class, please.
Proton dengan proton, artinya proton dengan proton tetangganya. Kalau dia ada korelasi, maka akan terlihat di spektrum. Dua karbon lebih jauh, jadi dari proton ke karbon yang mengikatnya, ke karbon tetangganya, dan ke karbon yang di sebelahnya lagi.
Keseluruhan workshop ini ya tentu sangat membantu bagi saya ya Pak. Walaupun saya sebenarnya di bioteknologi, basicnya biochemistry, adalah mungkin akan kesana juga untuk mengetahui senyawa kimia yang kita ingin kembangkan.
Iya betul, pas. Oke, silakan on kembali. Setelah itu bikin range-nya.
Ini nggak mau. Tapi kalau ini mau.
Karbon mau ya? Oke.
Detection, close, 20 display.
Oke. Coba HCMBC, Bu.
Okay, that's the video of our NMR intensive class. So for further explanation, please let me explain it in bahasa from now on. Oke, jadi sekarang saya ingin menjelaskan sedikit mengenai salah satu program yang dilakukan di PUI Nutraceutical. Salah satunya adalah kelas intensif NMR. Ini sudah berlangsung, ini kita buka sekarang untuk yang keempat kalinya ya. Sebelumnya telah berlangsung 3 kali pertemuan, 3 kali intensif kelas, dan sekarang kita buka untuk kelas yang keempat. Jadi untuk 3 kelas yang dibuka sebelumnya sudah lebih dari 100 peserta join, dan untuk register bagi teman-teman dan kolega yang ingin join dan register ataupun yang ingin tanya-tanya juga mengenai kelas intensif NMR ini, bisa register di link berikut, sudah ada di slide ya, bit.ly/4THNMR. Also kita juga dari sini bisa dapat free mengenai ebook NMR database of 2020, kemudian dapat software juga, kemudian NMR raw data untuk praktis, untuk latihan ya. Kemudian untuk info lebih lanjut bisa kontak juga di nomor berikut seperti yang di poster. Admin kita yaitu Nanda di nomor yang tertera di slide juga. Next. Operator, next please. Ya, jadi untuk pemateri akan ada 2 orang ya, akan dibuka 2 kelas. Nanti lebih lanjut ketika teman-teman Bapak dan Ibu telah mengisi link untuk register. Nanti akan dihubungi oleh panitia mengenai informasi lebih lanjut, dan kalau misalnya dari Bapak dan Ibu sekalian ada yang ingin ditanyakan, bisa langsung ditanyakan kepada admin yang bersangkutan. Untuk pemateri di sini kita ada terdiri 2 orang, Bapak Andi Rifta, moderator kita pada webinar hari ini. Kemudian ada Pak Laode juga. Untuk expertise-nya mirip-mirip ya. Jadi makanya jadi pemateri pada kelas intensif NMR kali. Oke, mohon dicatat ya untuk link registrasinya sekali lagi di bit.ly/4THNMR. Bisa langsung redirect ke form registrasinya. Langsung aja untuk registrasi bagi yang berminat, dan pun yang untuk tanya-tanya juga silahkan ya. Nanti langsung kontak aja adminnya, nomornya tadi Sarah. Nanti juga akan ada di link registrasinya juga untuk informasi adminnya. Oke, gitu aja ya, saya kembali lagi speak in English for official close. Okay, so once again, I would like to say thank you once again for all the speakers. Associate Professor Dr. Syarul Natakain Baharum, Dr. Arif Izairi Zamani, and Dr. Nizar Rapiana for your time to give such an insightful knowledge. I also want to say thank you for sponsorship Berka Niaga Medica, And the end of today's webinar, and again on the next webinar. Goodbye, stay healthy, and have a nice day.
Oke, thank you Dr. Ain. Makasih. Dr. Arif, makasih banyak.
Okay, thank you very
Pak
much.
Nizar.
Makasih Pak.
Assalamualaikum.
Thank you for all.
everyone
Saya
for
kontak balik.
hosting this great event. Yeah.
Oke,
Thank you.
Assalamualaikum.
Thank you.
Kolaborasi nanti ya.
Ya, baik, Dr. Agus.
Dr. Ain siap-siap tuh. Sarah, bagi tahu.
Boleh kolaborasi dengan
Besar juga.
I'm leaving. Bye. Assalamualaikum.
Salam. Terima kasih semua.
Stay safe everyone. Jaga diri ya.
ya.
Bye-bye.
Do you recall when we were young, running from all things at once, without thinking twice? And I knew it would catch up, and that we would be the ones left behind. The stories I've been told, they never seem to leave my mind. And this road that I am on, I gotta stay here for some time. Time is gone and I grew up. I somehow made it through without losing sight. And I still wonder where you are, and if you found a way out from the dark. The stories I've been told, they never seem to leave my mind. gotta stay here for some time. Bruises, they come and they go, and we have to try and keep up, 'cause this life's so much more. Ooh, and I still wonder where you are. Ooh, if you found a way out, come seek me out.
I did a lot of bad things, and never did I give in. Just 'cause the thought of being vulnerable made me feel less than I am. I have never felt home till you entered my door.